All right. Again, I want to take a break from our lab kit to look at another experiment, and that's the classic elephant toothpaste. And since we're doing the classic elephant toothpaste, the only thing I have to say to you is to write down BABAR. B-A-B-A-R. BABAR. A catalyst is anything that's the subject of this lab focuses around the science of chemical kinetics. Just like cars have speed, chemical reactions occur at different speeds. These speeds can be anything from picoseconds to millennium. So there's a wide range of the time span in which a chemical reaction can occur. The study of the speed at which chemical reactions referred to, the study of the speed at which chemical reactions occur is called chemical kinetics. I got that out. I'm so proud of myself. All right. If you were to study chemical kinetics, your goal is more than likely to make the chemical reaction go faster. There's big money in industry in making chemical reactions that normally occur very slowly, making them occur very fast. The way to make a chemical reaction to go faster is often to add what's known as a catalyst. A catalyst is a substance that helps a chemical reaction go faster, but it isn't consumed in the reaction. A chemical reaction is any substance that helps a chemical reaction go faster, but isn't used or consumed in the reaction. Nearly everything we use in our daily life was produced using a catalyst. Remember, I said they're really important to industry, right? Bread, cheese, plastics, and paper are all made using catalysts. Our body could not function without biological catalysts. Biological catalysts are given a special name. They're referred to as enzymes. So when you hear people talking about enzymes, they're talking about biological catalysts, chemicals inside your body that help your body perform processes faster. Matter of fact, they often help reactions that would occur too slowly to sustain our life without the presence of that enzyme or without the presence of that biological catalyst. Do you have a bottle of hydrogen peroxide at home? Have you ever thought about why hydrogen peroxide's always sold in a brown bottle? Well, let's take a look. Hydrogen peroxide has a chemical formula that's very close to water. We know that water is H2O, while hydrogen peroxide is H2O2, which brings up my favorite joke of all time, and I've probably told it a million times before. A chemist walks into a bar and asks the waiter for a glass of H2O. The friend, not wanting to be outdone by the chemist, decides that he'll play it cool too and tells the waiter, "I'd like a glass of H2O2, please." The waiter brings each of them their glasses. The friend takes a sip and collapses over dead because the waiter brought him H2O2, or hydrogen peroxide. Now, the hydrogen peroxide that you use for cleaning wounds at home is a paltry 3% concentration, or less. Probably less, because it was 3% when it leaves the factory. And as we'll talk about, hydrogen peroxide decomposes, for lack of a better term, very, very rapidly. Now, We know that hydrogen peroxide can bleach hair, but to bleach hair, you use a much stronger hydrogen peroxide concentration. You use a concentration of hydrogen peroxide that's in the neighborhood of 12%. 12% hydrogen peroxide is enough to bleach your hair. 30% hydrogen peroxide, which is what we have in our laboratory, is enough to strip the skin from your bones. So hydrogen peroxide is actually a very dangerous chemical in large enough concentrations. Obviously, the less than 3% that you have around the house is a relatively safe concentration to work with, but it is strong enough to clean wounds. Now, hydrogen peroxide doesn't like having that extra oxygen, and that's the reason it's kept in a brown bottle. Just a second, I'm trying to get my sheet of paper ready. Hydrogen peroxide doesn't like having that extra oxygen, and over time, two hydrogen peroxide molecules will team up and decompose to form two water molecules and oxygen gas. The type we breathe. Light is one of the things that can speed up the rate of this reaction. Because sunlight speeds up this reaction, we keep hydrogen peroxide in a brown bottle to protect it from light. There are also chemical catalysts that can speed up this chemical reaction, and that's what we're going to look at in today's experiment. We're actually going to use a biological catalyst or an enzyme to speed up the decomposition of hydrogen peroxide. Make a note of Horton. Make a note of Horton. H-O-R-T-O-N. Normally, the breakdown of hydrogen peroxide occurs very slowly. If we were just to set that hydrogen peroxide out in a glass on the kitchen table and we were to watch it, we probably wouldn't see any bubbles of oxygen. The rate of reaction would be too slow for us to really observe visually with our naked eyes. So what we're going to do is we're going to add a catalyst to speed up the reaction. Yeast, the type you cook with in the store, yeast, actually contains a catalyst that speeds up the reaction. The catalyst found in yeast is actually an enzyme named catalase. Now, oxygen is a gas, and it's a clear gas, and it's odorless. So it's kind of hard for us to tell when it's there. So we're going to have to add an indicator to this experiment. The indicator we're going to use for this experiment is going to be dishwashing detergent. Dishwashing detergent responds to the presence of a gas. How does dishwashing detergent respond to the presence of a gas? By blowing bubbles, right? So that's going to serve as our indicator in today's lab. You're going to do elephant toothpaste, the safe at home version, using the instructions provided in the link. Couple things that you need to be aware of and need to make sure you do, and I'm sure you were going to do this even before I told you to. You were going to wear goggles. Second, Second, use no more than 3% hydrogen peroxide. In other words, go to Walgreens, go to Kroger's, buy 3% hydrogen peroxide from the first aid section. You'll be able to check the back and see that it's 3%. If you turn it over and look at your label, you'll see hydrogen peroxide 3%. Do not use more than 3%. You'll find places online telling you to go to beauty salons and whatnot. Do not use more than 3%. It gets very dangerous when you use more than 3%. I had to use 30% hydrogen peroxide in the laboratory procedure for one of the parts of my PhD dissertation. I got the worst burns, serious chemical burns to my skin, serious skin damage. You can do yourself serious damage if you use more than 3%. So don't go on Amazon or anything like that and get fancy. Use 3% from the first aid section. Videotape. Realize this is messy and that food coloring will go everywhere, potentially, if this experiment goes right for you. So you're going to want to use a kitchen pan or something else to hold your bottle with. All right? That's all I really have for you, except why don't you write down Dumbo? Write down Dumbo. Because I've seen an elephant fly. Here are 3 questions to think about while you perform the experiment. Did the reaction occur fast enough for you to see? What caused the bubbles you're about to observe? And were we seeing the effects of the reaction or the effects the reaction had on the indicator?